Next topic, code value and complexity of genome. Code analysis, a technique based on the principles of DNA reassociation kinetics, is a biochemical technique that measures how much repetitive DNA is in DNA samples such as a genome. It is used to study genome structure and organization and has also been used to simplify the sequencing of genomes that contain large amounts of repetitive sequences. So, code analysis, a technique used on the principles of DNA reassociation kinetics, is a biochemical technique that measures how much repetitive DNA is in a DNA sample, such as a genome. It is used to study genome structure and organization and has also been used to simplify the sequencing of genomes that contain large amounts of repetitive sequence. The procedure involves heating a sample of genomic DNA until it denatures into the single-stranded form and then slowly cooling it so the strands can pair back together. While the sample is cooling, measurements are taken of how much of the DNA is base paired at each temperature. The amount of single and double-stranded DNA is measured by rapidly diluting the sample, which shows and slows reassociation, and then binding the DNA to a hydroxyl pyrrolate solution column. The procedure involves heating a sample of genomic DNA until it denatures into the single-stranded form and then slowly cooling it. So the strands can pair back together. While the sample is cooling, measurements are taken of how much of the DNA is base paired at each temperature. The amount of single and double-stranded DNA is measured by rapidly diluting the sample, which slows reassociation, and then binding the DNA to a hydroxypyrrolate column. The column is first washed with a low concentration of sodium phosphate buffer, which elutes the single-stranded DNA, and then with high concentrations of phosphate, which elutes the double-stranded DNA. The amount of DNA in these two solutions is then measured using a spectrophotometer. Repetitive sequence, single copy sequence. Repetitive DNA sequences renature at lower code values than single copy sequences. Analysis. Since a sequence of single-stranded DNA needs to find its complementary strand to reform a double helix, common sequences denature more rapidly than rare sequences. Indeed, the rate at which a sequence will reassociate is proportional to the number of copies of that sequence in the DNA sample. A sample with a highly repetitive sequence will renature rapidly, while complex sequences will renature slowly. However, instead of simply measuring the percentage of double-stranded DNA versus time, the amount of renaturation is measured relative to coat value. The coat value is the product of CO, the initial concentration of DNA, T, time in seconds, and a constant that depends on the concentration of cations in the buffer. So, initial concentration of DNA, cations in the buffer, and time. Repetitive DNA will renature at low coat values, while complex and unique DNA sequences will renature at high coat values. The fast renaturation of the repetitive DNA is because of the availability of numerous complementary sequences. DNA denaturation is the separation of a double strand into two single strands, which occurs when the hydrogen bonds between the strands are broken. Denaturation and renaturation, DNA helix, two strands separated. Denaturation and renaturation of DNA, double helix unwind when DNA is denatured, can be reformed with slow cooling and annealing. Native double helix strands unwind, separate strands, double helix reformed. Renaturation. Native state, single stranded denatured state, renatured state. It is a technique for measuring the complexity size of DNA or genome. The technique was developed by Roy Britton and Eric Davidson in 1960. The technique is based on the principle of DNA renaturation kinetics. The rate of renaturation is directly proportional to the number of times the sequences are present in the genome. Given enough time, all DNA that is denatured will reassociate or reanneal in a given DNA sample. The more the repetitive sequence, the less will be the time taken for renaturation. Procedure. The process involves denaturation of DNA by heating. and allowed to reanneal by cooling. The renaturation of DNA is assessed stereoscopically. Large DNA molecule take longer time to reanneal. What is coat value? The renaturation depends on the following factors: DNA concentration, reassociation temperature, cation concentration, and viscosity. Coat DNA concentration moles per liter into renaturation time in seconds x buffer factor that accounts for the effect of cation on the speed of renaturation. Coat CO concentration of DNA at T, time taken for sequences. Low coat value indicates more number of repetitive. High coat value indicates more number of unique sequences or less number of repetitive sequences. How to calculate coat value? Coat is equal to DNA concentration moles per liter into renaturation time in second into buffer factor. That accounts for the effect of cations in the speed of renaturation. Nucleotide concentration 0.050 m, renaturation time 344 seconds, coat value like this. Buffer factor 0.5 molar SPB. Percentage single stranded DNA, highly repetitive, middle repetitive, single copy. The x-axis is a log scale of the product of the initial concentration of DNA in moles liter multiplied by length of time the reaction proceeded in seconds. The designation for this value is coat and is called the coat value. The curve itself is called a coat curve. Genomic complexity. Britton and Davidson defined the relative amounts of repeated and unique or single copy DNA sequences in an organism's genome as its genomic complexity. Thus, prokaryotic genomes have a lower genomic complexity than eukaryotes. Then application of coat curve analysis, understanding genome size and complexity, understanding complexity of sequences, understanding relative proportion of single copy and repetitive sequences. So, applications, understanding genome size and complexity, understanding complexity of sequences, understanding relative proportion of single copy and repetitive sequences. Next topic, junk DNA, satellite DNA and selfish DNA. Non-coding DNA. Non-coding DNA. Non-coding DNA sequences are components of an organism's DNA that do not encode protein sequences. Some non-coding DNA is transcribed into functional non-coding RNA molecules, transfer RNA, ribosomal RNA, and regulatory RNAs. Other functions of non-coding DNA include the transcriptional and translational regulation of protein coding sequences, scaffold attachment regions. Other functions of non-coding DNA include the transcriptional and translational regulation of protein coding sequences, scaffold attachment regions, origins of DNA replication, centromere and telomere. Its RNA counterpart is non-coding RNA. Junk DNA. The amount of non-coding DNA varies greatly among species. Often only a small percentage of the genome is responsible for coding proteins, but an increasing percentage is being shown to have regulatory functions. When there is much non-coding DNA, a large proportion appears to have no biological function, as predicted in the 1960s. Since that time, this non-functional portion has controversially been called junk DNA. The term junk DNA became popular in the 1960s. According to Ryan Gregory, the nature of junk DNA was first discussed explicitly in 1972 by a genomic biologist, David Cummings, who applied the term to all non-coding DNA. The term was formalized that same year by Susumu Ohino, who noted that the mutational load from deleterious mutations placed an upper limit on the number of functional loci that could be expected given a typical mutation rate. Ohino hypothesized that mammal genomes could not have more than 300 loci under selection before the cost from the mutational load would cause an high decline in fitness and eventually extinction. This prediction remains robust, with the human genome containing approximately protein coding 20,000 genes. Another source of Ohno's theory was the observation that even closely related species can have widely orders of magnitude different genome sizes, which had been dubbed the C-value paradox in 1971. Since the late 70s, it has become apparent that the majority of non-coding DNA in large genomes finds its origin in the selfish amplification of transposable elements, of which W. Ford Doolittle and Carmen Sapienza in 1980 wrote in the journal Nature. The term occurs mainly in popular science and in a colloquial way in scientific publications, and it has been suggested that its connotations can be delayed interest in the biological functions of non-coding DNA. Some evidence indicates that some junk DNA frequencies are sources for future functional activity in evolution through exaptation of originally selfish or non-functional DNA. The amount of total genomic DNA varies widely between organisms, and the proportion of coding and non-coding DNA within these genomes varies greatly as well. For example, it was originally suggested that over 98% of human genome does not encode protein sequences, including most sequences within introns and most intergenic DNA, while 20% of typical prokaryote genome is non-coding. The amount of junk DNA can be expected to depend on the rate of amplification of these elements and the rate at which non-functional DNA is lost. Types of non-coding DNA, regulatory genes and introns. Non-coding DNA is also found in the form of cis and trans regulatory elements that modulate gene transcription. They are found either within introns or in untranslated regions at the 5' or 3' ends of the gene. Cis and trans refer to their location within and between chromosomes respectively. An intron is a stretch of non-coding DNA incorporated into the gene sequence itself. Introns are therefore non-coding DNA by definition and are transcribed into the preliminary messenger RNA molecule, but are then removed to give rise to the mature form. They may play regulatory roles in controlling the activity of tRNA and rRNA as well as of the protein encoding segments of codons. However, most introns are not functional. All genes have a regulatory site called a promoter sequence, which is a non-coding DNA segment that is bound by proteins involved in the process of transcription. Such promoter sequences do not give rise to any part of the final protein, but facilitate the transcription of a particular gene and are usually found upstream of the coding region. Enhancer sequence also influence the likelihood that a gene will be transcribed. Proteins that activate transcription bind to this short sequences. On the other hand, inhibitory sequences or silencers may also be present, which are open to binding by inhibitory proteins that repress or reduce the chances of transcription. Silencer sequences are found in a little distance away from the gene they regulate, either before or after it. Super enhancers are clusters of enhancer sequence bound together by physical or functional association and which are tied to regulation of genes that are vital for the cell's identity, such as the transcription factors that determine the type and image of the cell. Both types of regulatory elements may be present in some genes which require a high degree of regulation. Then pseudogenes. Another type of non-coding DNA is the pseudogene, which is a DNA sequence that resembles an existing gene but is non-functional. These are thought to be the result of mutations in functional genes that prevent their forming of functional proteins or inhibit their transcription. They could also arise as a result of retrotransposition. Most of them seem to be non-functional. Some viral infections may also result in non-coding DNA as the result of reverse transcription. This process describes what happens when RNA-carrying virus like HIV infects a cell. It copies its RNA in the form of DNA onto the host DNA so that it can make the host cell carry out the various operations required to replicate and proliferate. These virally derived DNA sequences may undergo mutations later, which lead to their inactivation, forming pseudogenes. Then transposons. Another specialized type of non-coding DNA is the transposon, a mobile genetic element that can change its location in the genome. By shifting its location, it may correct a mutation or induce one. In either case, it changes the size of the cell's genome. Transposable elements make up the major part of the non-coding DNA. This include lines, SINE, satellite DNA, and VNTR. Lines or long interspaced elements are moderately repetitive non-coding regions, possibly derived from virus. SINE, short Interspaced elements are highly repetitive non-functional regions that may be the result of reverse transcription of RNA. Telomeres and satellite DNA. Telomeres are segments of repeating nucleotides forming specialized DNA segments found at the ends of all chromosomes. These are important in preserving the chromosome's structural integrity during the process of DNA replication by keeping the ends from being degraded. Satellite DNA is a term used for tandemly repetitive DNA regions clustered in an area. This type of non-coding DNA is found in centromeres, the viral structure that link the members of a chromosome pair during cell division. It is also present in the form of heterochromatin, a densely packed form of DNA that regulates gene activity as well as preserves chromosome structure. VNTRs, or variable number of tandem repeats, are repetitive elements but shorter than seen with satellite DNA. Functions of non-coding DNA. Some non-coding DNA sequences must have some important biological function. This is indicated by comparative genomics studies that report highly conserved regions of non-coding DNA, sometimes on timescales of hundreds of millions of years. This implies that these non-coding regions are under strong evolutionary pressure and positive selection. Non-coding genetic polymorphisms play a role in infectious disease susceptibility such as hepatitis C. Moreover, non-coding genetic polymorphisms contribute to susceptibility to Ewing sarcoma and aggressively pediatric bone cancer. Ewing sarcoma. Non-coding DNA may perhaps serve to decrease the probability of gene disruption during chromosomal crossover. Some specific sequences of non-coding DNA may be feature essential to chromosome structure, centromere function, and recognition of homologous chromosome during meiosis. Then regulating gene expression. Some non-coding DNA sequences determine the expression levels of various genes. Both those that are transcribed to proteins and those that transcribe themselves are involved in gene regulation. Some non-coding DNA sequences determine where transcription factors attach. A transcription factor is a protein that binds to specific non-coding DNA sequences, thereby controlling the flow or transcription of genetic information from DNA to mRNA. Then operators. An operator is a segment of DNA to which a repressor binds. A repressor is a DNA binding protein that regulates the expression of one or more genes by binding to the operator and blocking the attachment of RNA polymerase to the promoter, thus preventing transcription of the genes. This blocking of expression is called repression. Then enhancers. An enhancer is a short region of DNA that can be bound with proteins, trans-acting factors, much like a set of transcription factors to enhance transcription levels of genes in a gene cluster. Then silencers. A silencer is a region of DNA that inactivates gene expression when bound by a regulatory protein. It functions in a very similar way as enhancer, only differing in the inactivation of genes. Then promoters. A promoter is a region of DNA that facilitates transcription of a particular gene with a transcription factor binds to it. Promoters are typically located near the genes they regulate and upstream of them. Insulators. A genetic insulator is a boundary element that plays two distinct roles in gene expression, either as an enhancer, blocking code, or rarely as a barrier against condensed chromatin. An insulator in a DNA sequence is comparable to a linguistic word divider such as a comma in a sentence, because the insulator indicates where an enhanced or repressed sequence ends. That is insulator. Then applications, long-range correlations. The statistical distinction between coding and non-coding DNA sequences has been found. It has been observed that nucleotides and non-coding DNA sequences display long-range power-law correlations, while coding sequences do not. Evolution. Shared sequences of apparently non-functional DNA are a major line of evidence of common descent. Pseudogene sequences appear to accumulate mutations more rapidly than coding sequence due to loss of selective pressure. This allows for the creation of mutant alleles that incorporate new functions that may be favored by natural selection. Thus, pseudogenes can serve as raw material for evolution and can be considered protogenes. Then forensic anthropology. Police sometimes gather DNA as evidence for purposes of forensic identification. Then satellite DNA. Satellite DNA consists of a very large array of tandemly repeating non-coding DNA. Satellite DNA is the main component of functional centromere and form the main structural constituent of heterochromatin. The name satellite DNA refers to the phenomenon that repetitions of a short DNA sequence tend to produce a different frequency of the base adenine, cytosine, guanine, and thymine, and thus have a different density from the bulk DNA such that they form a second or satellite band when genomic DNA is separated on a density gradient. Sequences with a great ratio of A plus T display a lower density, while those with a greater ratio of G plus C display a higher density than the bulk genomic DNA. Satellite DNA, together with mini-satellite and microsatellite DNA, constitute the tandem repeats. Microsatellites are thought to have originated by polymerase slippage during DNA replication. This comes from the observation that microsatellite alleles usually are length polymorphisms. Specifically, the length differences observed between microsatellite alleles are generally multiples of repeat unit length. Microsatellite expansion, trinucleotide repeat expansion, is often found in transcription units. Often the base pair repetition will disrupt proper protein synthesis, leading to diseases such as myotonic dystrophy. Then function. Satellite DNA contribute to the essential processes of formation of crucial chromosome structures, heterochromatin establishment, dosage compensation, reproducible isolation, genome stability and development. Genomic abundance of satellites is under stabilizing selection owing to their role in the maintenance of vital regions of the genome, centromere, pericentromeric region, and telomerase. Many satellites are transcribed with the generation of long or small non-coding RNAs. Misregulation of their expression is found to lead to various defects in the maintenance of genomic architecture, chromosome segregation, and gametogenesis. Next, selfish DNA. Selfish genetic elements or selfish DNA are genetic segments that can enhance their own transmission at the expense of other genes in the genome, even if it has no positive or negative effect on the organismal fitness. Genomes have traditionally been viewed as cohesive units, with genes acting together to improve the fitness of the organism. However, when genes have some control over their own transmission, the rules can change. And so, just like all social groups, genomes are vulnerable to selfish behavior by their parts. Inspired by the gene-centered use of evolution, popularized by George Williams and Richard Dawkins, two papers were published back-to-back in Nature in 1980 by Leslie Orgel and Francis Crick, and by Ford Doolittle and Carmen Sapienza, introducing the concept of selfish genetic elements, at the time called selfish DNA, to the wider specific scientific community. Both papers emphasized that genes can spread in a population regardless of their effect on organismal fitness, as long as they have a transmission advantage. Selfish genetic elements have now been transcribed in most groups of organisms, and they demonstrate a remarkable diversity in the ways by which they promote their own transmission. Through long diminished as genetic curiosities with little relevance for evolution, they are now recognized to affect a wide swath of biological processes ranging from genome size and architecture to speciation. Examples of selfish gene segregating distorters. Some selfish genetic elements manipulate the genetic transmission process to their own advantage and so end up being overrepresented in the gamete. Such distortion can occur in various ways, and the umbrella term that encompasses all of them is segregation distortion. Some elements can preferentially be transmitted in egg cells as opposed to polar bodies during meiosis, where only the former will be fertilized and transmitted to the next generation. Any gene that can manipulate the odds of ending up in the egg rather than the polar body will have a transmission advantage and will increase the frequency in a population. Then homing endonuclease, a phenomenon closely related to segregation distortion, is homing endonucleases. These are enzymes that cut DNA in a sequence specific way, and those cuts, generally double strand breaks, are then healed by the regular DNA repair machinery. Homing endonucleases insert themselves into the genome at the site of homologous to their first insertion site, resulting in a conversion of a heterozygote into a homozygote bearing a copy of the forming endonuclease on both homologous chromosomes. Transposable elements. Transposable elements including a wide variety of DNA sequences that all have the ability to move to new locations in the genome of their host. B chromosomes. B chromosomes refer to chromosomes that are not required for the viability or fertility of the organism but exist in addition to the normal A set. They persist in the population and accumulate because they have the ability to propagate in their transmission independently of the A chromosome. They often vary in copy number between individuals of the same species. Then selfish mitochondria. Genomic conflicts often arise because not all genes are inherited in the same way. Probably the best example of this is the conflict between uniparentally, usually but not always, maternally inherited mitochondrial and biparentally inherited nuclear genes. Genomic imprinting. Another sort of conflict that genomes face is that between the mother and father competing for control of gene expression in the offspring, including the complete silencing of one parental allele. Due to differences in methylation status of gametes, there is an inherent asymmetry of the maternal and paternal genomes that can be used to drive a differential parent of origin expression. This results in violation of Mendel's rules at the level of expression, not transmission. But if the gene expression affects fitness, it can amount to a similar end result. Imprinting seems like a maladaptive phenomenon since it essentially means giving up diploidy and heterozygotes for one distinctive allele are in trouble if the active allele is the one that is silenced. Several human diseases such as Prader-Willi and Angelman syndrome are associated with the defects in imprinted genes. Then Guillain-Barré gene. A Guillain-Barré gene is a gene that has an ability to recognize copies of itself in other individuals and then makes its carrier act preferentially towards such individuals. The name itself comes from thorough experiment first presented by Billy Hamilton and then it was developed and given its current name by Richard Dawkins in Satellite Selfish DNA. Application: cytoplasmic male sterility in plant breeding. A common problem for plant breeders is unwanted self-fertilization. This is particularly a problem when breeders try to cross different strains to create a new hybrid strain. One way to avoid this is manual emasculation, physical removing anthers to render the individual male sterility. Cytoplasmic male sterility offers an alternative to this laborious exercise. Breeders cross a strain that carries a cytoplasmic male sterility mutation in a strain that does not, the laser acting as the pollen donor. Then piggyback vectors. While many transposable elements seems to do no good for the host, some transposable elements have been tamed by molecular biologists so that the elements can be made to insert and excise at the will of the scientists. Such elements are especially useful for doing genetic manipulations like inserting foreign DNA into the genomes of a variety of organisms. One excellent example of this is piggyback, a transposable element that can efficiently move between cloning vectors and chromosomes using a cut and paste mechanism. CRISPR gene drive and homing endonuclease systems. CRISPR allows the reconstruction of artificial homing endonuclease, where the reconstruct produces guide RNAs that cut the target gene and homologous flanking sequence then allow insertion of the same construct harboring the Cas9 gene and the guide RNAs. Such gene drives ought to have the ability to rapidly spread in a population. See gene drive systems, and one practical application of such a system that has been proposed is to apply it to a pest population, greatly reducing its numbers or even driving it extinct.